Slide 1. Title: Genetics and Heredity, the Blueprint of Life. Subtitle: Understanding Inheritance, DNA, and Genetic Variation. Introduction. Welcome, everyone, to our exploration of genetics and heredity, the cornerstones of life's diversity. In this presentation, we will unravel the intricate world of genes, chromosomes, and DNA, gaining insights into how genetic information is passed from one generation to the next. Understanding these fundamental concepts is essential not only for biology enthusiasts but for anyone curious about the essence of life itself. Objective. Our journey begins with an introduction to the fascinating realm of genetics. We will delve into Mendel's laws of inheritance, exploring how traits are inherited from parents to offspring. Moving forward, we will dissect the structure and role of DNA, the blueprint of life, and comprehend the significance of accurate DNA replication. Throughout our exploration, we will touch upon genetic variations, mutations, and their implications, shedding light on the complexity of genetics in the natural world. Importance. Why study genetics? Genetics not only explains the inheritance of physical traits but also underpins our understanding of diseases, evolution, and biodiversity. It serves as the foundation for medical research, biotechnology, and genetic engineering, leading to breakthroughs in healthcare and agriculture. By comprehending genetics, we unlock the secrets of life, paving the way for scientific advancements that shape our future. As we embark on this genetic odyssey, keep in mind that every living being is a product of its genetic makeup. Together, let's unravel the mysteries encoded within our DNA, appreciating the elegance of nature's design. So, fasten your seat belts, and let's journey into the intricate world of genetics and heredity, where the blueprint of life awaits our exploration. Slide 2. Introduction to Genetics. Definition and Significance. Genetics, the study of heredity and variation in living organisms, forms the backbone of modern biology. It delves into the mechanisms of trait inheritance, allowing us to trace the origins of specific traits within populations. Understanding genetics is crucial not only for evolutionary studies but also for diagnosing genetic disorders, creating genetically modified organisms, and advancing medical treatments. Historical context. The roots of genetics can be traced back to Gregor Mendel, whose pioneering work with pea plants laid the foundation for our understanding of hereditary patterns. His laws of inheritance provided a framework for subsequent genetic research, demonstrating the segregation and independent assortment of traits. Since Mendel's time, genetics has evolved significantly, leading to groundbreaking discoveries such as the structure of DNA, elucidated by Watson and Crick in 1953. Scope of genetics. Genetics encompasses a broad spectrum of topics, including molecular genetics, studying DNA and genes at the molecular level, population genetics, examining genetic variation within populations, and genomics, analyzing entire genomes. With the advent of advanced technologies like CRISPR-Cas9, genetics has entered an era of unprecedented possibilities, allowing us to edit genes and potentially cure genetic diseases. Challenges and ethical considerations. While genetics holds immense potential, it also presents ethical dilemmas, particularly concerning genetic engineering and cloning. As we explore the intricacies of genetics, we must navigate the ethical challenges, ensuring that scientific advancements are used responsibly and for the greater good of humanity. Slide 3. Mendel's Laws of Inheritance. Law of Segregation. Mendel's law of segregation states that each individual has two alleles for each gene, one inherited from each parent. During gamete formation, these alleles segregate, meaning that each gamete carries only one allele for each trait. When two gametes fuse during fertilization, the offspring's genotype is determined, influencing their observable traits, or phenotype. Law of Independent Assortment. The law of independent assortment asserts that the alleles for different traits segregate independently of one another during the formation of gametes. In other words, the inheritance of one trait does not affect the inheritance of another trait. This law explains the diversity of traits within populations and underpins our understanding of genetic variation. Mendelian inheritance patterns. Mendel's laws give rise to specific inheritance patterns, including dominant recessive inheritance, codominance, and incomplete dominance. Dominant recessive inheritance occurs when one allele masks the effect of another, dominant, while codominance results in both alleles being expressed equally. Incomplete dominance leads to a blending of traits, creating an intermediate phenotype in heterozygous individuals. Applications and exceptions. Mendel's laws provide a fundamental framework for understanding inheritance, but exceptions do exist, such as traits influenced by multiple genes, polygenic traits, or those influenced by both genes and the environment, multifactorial traits. Despite exceptions, Mendel's work laid the groundwork for modern genetics, guiding our exploration of the genetic basis of traits and diseases. Slide 4. DNA, the genetic material. Structure of DNA. DNA, or deoxyribonucleic acid, is a molecule that carries the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms and many viruses. It has a double helix structure, composed of nucleotides. Each nucleotide consists of a phosphate group, a sugar molecule, deoxyribose, and one of four nitrogenous bases, adenine, A, guanine, G, cytosine, C, or thymine, T. The sequence of these bases encodes genetic information. Role of DNA. DNA serves as the genetic material because it can store information in the sequence of its nucleotides and replicate this information accurately. During cell division, DNA is replicated, ensuring that each daughter cell receives a complete and accurate set of genetic instructions. Additionally, DNA encodes genes, which are segments of DNA that provide the instructions for making specific proteins. Proteins, in turn, govern various biological functions in cells and organisms. Genetic code and proteins. The sequence of nucleotides in DNA forms a genetic code, where each set of 3 nucleotides, a codon, corresponds to a specific amino acid. Amino acids are the building blocks of proteins. The process of protein synthesis involves decoding the genetic information in DNA and assembling the corresponding amino acids into proteins. This intricate process is essential for the structure and functioning of cells. Significance. Understanding the structure and function of DNA is pivotal in genetics. It not only illuminates the mechanism of inheritance but also provides insights into genetic variations, mutations, and the molecular basis of diseases. DNA research has led to revolutionary discoveries in fields like medicine, forensics, and biotechnology, shaping our understanding of life and enabling scientific advancements. Slide 5. DNA replication. Process of DNA replication. DNA replication is a semi-conservative process where a DNA molecule makes an identical copy of itself. It occurs during the cell cycle, ensuring that genetic information is passed from parent cells to daughter cells. The process starts with the separation of the DNA strands, facilitated by enzymes called helicases. Each separated strand serves as a template for the synthesis of a new complementary strand. Enzymes called DNA polymerases add nucleotides to the growing strands, following the base pairing rules, AT, CG. Finally, ligases seal any gaps, forming two identical DNA molecules. Accuracy and proofreading. DNA replication is highly accurate, thanks to the proofreading mechanisms of DNA polymerases. These enzymes can recognize and correct errors that occur during replication, maintaining the integrity of the genetic information. Despite this accuracy, occasional mutations can still happen, leading to genetic diversity, which is essential for evolution and adaptation. Importance of accurate replication. Accurate DNA replication is crucial because any errors or mutations in the genetic code can result in genetic disorders or diseases, including cancer. Cells have intricate repair mechanisms to fix DNA damage and ensure accurate replication. Understanding these processes not only sheds light on genetic stability but also informs cancer research and therapies targeting aberrant cell division. Replication in cell division. In multicellular organisms, DNA replication is a fundamental aspect of cell division. The accurate transmission of genetic information during replication ensures that daughter cells inherit the correct genetic instructions. Disruptions in the replication process can lead to cell death or uncontrolled cell growth, contributing to diseases like cancer. Slide 6. Genes and alleles. Definition of genes and alleles. Genes are specific sequences of DNA that encode instructions for building proteins. Each gene has a specific location on a chromosome. Alleles are different versions of a gene that can lead to different traits. For example, there are multiple alleles for the gene responsible for eye color, resulting in variations such as blue, green, or brown eyes. Alleles can be dominant or recessive, determining how they express in the phenotype. Dominant and recessive alleles. Dominant alleles mask the effects of recessive alleles when an individual has one dominant allele and one recessive allele, heterozygous. Only when an individual has two recessive alleles, homozygous recessive, will the recessive trait be expressed. Understanding dominant-recessive relationships is key to predicting the outcomes of genetic crosses and understanding the inheritance of specific traits. Genetic variation and alleles. Genetic variation within populations arises from differences in alleles. These variations can be beneficial, neutral, or harmful, depending on the environment. Natural selection acts on these variations, favoring alleles that confer advantages in specific environments. Over generations, this process leads to the adaptation of populations to their surroundings, enhancing their chances of survival and reproduction. Genetic disorders and alleles. Many genetic disorders are the result of mutations in specific alleles. In some cases, a mutated allele leads to the production of a non-functional or malfunctioning protein, disrupting normal cellular processes. Common genetic disorders, such as cystic fibrosis and sickle cell anemia, result from specific alleles that cause these conditions. Studying these alleles is essential for diagnosing and understanding genetic diseases. Slide 7. Genetic variation and mutations. Sources of genetic variation. Genetic variation in populations arises from several sources, including mutations, genetic recombination, and gene flow. Mutations are random changes in DNA sequences and can occur due to environmental factors, errors in DNA replication, or external agents like radiation or chemicals. Genetic recombination, which occurs during meiosis, shuffles alleles between homologous chromosomes, creating unique genetic combinations in offspring. Types of mutations. Mutations can be classified into various types, such as point mutations, changes in a single nucleotide, insertions, addition of nucleotides, and deletions, loss of nucleotides. Silent mutations do not alter the resulting protein, while missense and nonsense mutations change amino acids in the protein sequence. Frameshift mutations, caused by insertions or deletions, can lead to significant changes in the protein's structure and function. Impact of mutations. Mutations play a crucial role in evolution, providing the raw material for natural selection. Beneficial mutations enhance an organism's fitness and are more likely to be passed on to future generations. Harmful mutations can lead to genetic disorders or reduced fitness. Understanding the effects of mutations informs medical research, helping scientists develop treatments for genetic diseases and comprehend the genetic basis of cancer. Genetic diversity and adaptation. Genetic diversity resulting from mutations and genetic recombination is the foundation of adaptation and evolution. Populations with higher genetic diversity have a greater chance of adapting to changing environments. Genetic diversity also provides a reservoir of potential traits that can be advantageous in specific conditions, ensuring the survival of species in diverse ecological niches. Slide 8. Genetic disorders. Common genetic disorders. Genetic disorders are conditions caused by abnormalities in an individual's DNA. These disorders can be inherited from one or both parents and may affect physical or mental health. Common genetic disorders include cystic fibrosis, sickle cell anemia, Huntington's disease, and Down syndrome. Each disorder has unique genetic causes, symptoms, and inheritance patterns, making them crucial areas of study in medical genetics. Causes and symptoms. Genetic disorders can result from mutations in a single gene, monogenic disorders, or a combination of genetic and environmental factors, multifactorial disorders. The symptoms of genetic disorders vary widely and can impact various organ systems. Early diagnosis and genetic counseling are essential for managing these conditions, enabling affected individuals and their families to make informed decisions about their health care. Inheritance patterns. Genetic disorders follow specific inheritance patterns, including autosomal dominant, autosomal recessive, and X-linked inheritance. Autosomal dominant disorders occur when a person inherits a single copy of the defective gene from either parent. Autosomal recessive disorders require two copies of the defective gene, one from each parent, for the condition to manifest. X-linked disorders are caused by mutations in genes located on the X chromosome and predominantly affect males. Genetic counseling and treatment. Genetic counseling plays a vital role in helping individuals understand their risk of inherited disorders and make informed decisions about family planning. Advances in medical genetics have led to therapies like gene therapy, where defective genes are corrected or replaced, offering hope for the treatment of genetic disorders. Ongoing research in this field continues to expand our knowledge and improve the lives of individuals affected by these conditions. Slide 9. Genetic engineering and biotechnology. Definition of genetic engineering. Genetic engineering involves the manipulation of an organism's genes using biotechnological techniques. Scientists can insert, delete, or modify specific genes to achieve desired traits or outcomes. This technology has applications in medicine, agriculture, and industry, offering innovative solutions to various challenges and improving the quality of life. Applications in medicine. In medicine, genetic engineering has led to the development of gene therapies, where faulty genes are replaced or repaired to treat genetic disorders. This revolutionary approach holds promise for conditions like cystic fibrosis, muscular dystrophy, and certain types of cancer. Genetic engineering also enables the production of therapeutic proteins, such as insulin and growth hormones, used to treat various diseases. Applications in agriculture. Genetically modified organisms, GMOs, are crops or animals whose genetic material has been altered to enhance desirable traits. GMOs have been engineered to resist pests, tolerate harsh environmental conditions, and improve nutritional content. These modifications contribute to higher crop yields, reduced pesticide usage, and enhanced food security, addressing global challenges in agriculture. Applications in industry. Genetic engineering plays a crucial role in the production of enzymes, biofuels, and biodegradable plastics. Enzymes produced through genetic engineering processes are used in various industrial applications, including textile manufacturing and wastewater treatment. Additionally, genetically engineered microorganisms are employed to produce biofuels, contributing to sustainable energy solutions and reducing dependence on fossil fuels. Slide 10. Ethics of genetic engineering. Ethical considerations. The ethical implications of genetic engineering are multifaceted. Questions arise concerning the morality of altering an organism's genetic makeup, especially in the context of human gene editing. Ethical dilemmas include concerns about unintended consequences, environmental impact, and the potential for creating designer babies with specific traits. Striking a balance between scientific advancement and ethical responsibility is essential in the field of genetic engineering. Debate on genetic modification. The debate on genetic modification extends to agricultural practices. While GMOs have the potential to address food scarcity and enhance crop resilience, critics express concerns about their long-term effects on ecosystems, biodiversity, and human health. Regulations and guidelines are in place to ensure the safety of GMOs, yet public perception and ethical considerations continue to shape the discourse around their usage. Gene editing in humans. The ability to edit genes in humans raises profound ethical questions. While gene editing techniques like CRISPR-Cas9 offer possibilities for treating genetic diseases, They also present ethical challenges related to consent, accessibility, and equitable distribution of genetic therapies. International scientific communities are working to establish ethical frameworks and guidelines to govern human gene editing research and applications. Responsible innovation. The ethical discussion surrounding genetic engineering emphasizes the importance of responsible innovation. Scientists, policymakers, and society must collaborate to establish ethical boundaries, ensure transparency in research, and promote inclusivity in decision-making processes. By embracing ethical considerations, we can harness the potential of genetic engineering for the greater good while safeguarding ethical principles and human values. Slide 11, Human Genome Project. Overview of the Human Genome Project. The Human Genome Project, HGP, was an international research effort aimed at mapping and understanding all the genes of the human species. Completed in 2003, the project identified and mapped the approximately 20,000 to 25,000 genes in human DNA. The HGP provided a foundation for understanding the genetic basis of human traits, diseases, and evolution, opening avenues for genomic medicine and personalized healthcare. Impact on understanding human genetics. The HGP revolutionized our understanding of human genetics. By identifying genes associated with diseases and traits, scientists gained insights into the genetic underpinnings of conditions like cancer, diabetes, and neurological disorders. This knowledge paved the way for targeted therapies, diagnostic tests, and preventive measures, leading to advancements in precision medicine, genomic medicine, and personalized healthcare. Genomic medicine utilizes information from an individual's genome to guide medical decisions. It enables personalized treatment plans tailored to an individual's genetic makeup, improving the effectiveness of therapies and reducing adverse reactions. Genomic data also enhance disease risk prediction, enabling proactive interventions and preventive measures for at-risk individuals. Future prospects and research. The legacy of the Human Genome Project continues through ongoing research initiatives, exploring non-coding regions of the genome, epigenetic modifications, and gene-environment interactions. Advances in genomic technologies and data analysis techniques are accelerating our understanding of complex biological processes, fostering collaborations across disciplines, and driving innovations in healthcare and biomedical research. Slide 12, Conclusion and Future Prospects. Recap of key concepts. In our journey through the world of genetics and heredity, we have uncovered key concepts that form the blueprint of life. From Mendel's laws of inheritance to the structure and function of DNA, we have gained insights into the molecular basis of traits and genetic diversity. We have explored the role of genes, alleles, and mutations in shaping the diversity of life on Earth. Future advancements and breakthroughs. Our exploration doesn't end here. The field of genetics is dynamic and continually evolving. As technology advances, so does our understanding of the genetic code. Future breakthroughs may include precision gene therapies, the elucidation of the genetic basis of complex diseases, and innovations in personalized medicine. Genetic research holds the promise of enhancing our quality of life and addressing some of the most pressing challenges in healthcare and agriculture. The elegance of nature's design. Throughout our journey, we have marveled at the elegance of nature's design. The intricate processes of DNA replication, genetic variation, and natural selection have produced the astounding biodiversity we observe in the world around us. Understanding genetics is not only a scientific endeavor but an appreciation of the beauty and complexity of life. Your role in the genetic odyssey. As students of biology, you are at the forefront of unraveling the mysteries of genetics. Whether you choose careers in research, medicine, or conservation, your understanding of genetics will contribute to the advancement of science and the well-being of future generations. Embrace the excitement of discovery, ethical responsibility, and the potential to make a difference in the world through your knowledge of genetics. Closing remarks. In closing, genetics and heredity, the blueprint of life, is a remarkable journey into the very essence of life. Genetics touches every aspect of our existence, from the traits that make us unique to the diseases that challenge our health. The exploration of genetics is an ongoing endeavor that transcends disciplines and spans generations. As you continue your educational journey, remember the role genetics plays in the world and the potential it holds for the future. We encourage you to delve deeper into this fascinating field, ask questions, seek answers, and explore the boundless possibilities that genetics offers. In your pursuit of knowledge, consider the ethical implications of genetic research and the power of responsible innovation. Through your passion and commitment, you have the opportunity to contribute to the ever-evolving story of genetics, shaping the blueprint of life for generations to come. Thank you for joining us on this genetic odyssey. We wish you success in your studies and your future endeavors, and we look forward to witnessing the contributions you will make to the field of genetics and the broader world of science.